Let me start. Abdul started. Okay. So Abdul, what's our target on today? So should we take any of the topic from the component two? Now we have all organisms are related to their evolutionary history, adaptations for gas exchange, adaptations for transport, adaptations for.
No, you said that we will practice the cell cycle, more hard questions of cell cycle, right?
Oh yes, okay, fine.
According
We'll
to the
okay,
specification.
okay. As per it means like we'll get back to the component one.
Yeah.
Okay, in component one, it's the last topic here in component number six. Genetic
Yeah.
information is copied and passed on to daughter cells. All right.
Tomorrow I will prepare one of the topics from component two.
Component two, yeah. Okay, fine. But I think, Abdul, what we should do now, like somewhere, suppose that we have covered in component one, if I let me recall that. Um, I think cell membrane and transport, this is what we have discussed, right? In component one. Is this like the questions
Yeah.
from this topic?
Yeah. Yeah.
And what's about
Yes.
cell structure and organization? Have you worked on this or not?
Uh,
Cell structure and
the
organization.
I do,
I think not
yeah.
because
No. The
because we.
first, yeah, the
Hmm.
very first one is very easy. It's fine.
Yes, that's fine, but at least we should take some hard questions for these two topics as well. That's fine. I
So
mean, if you
tomorrow,
think like.
so tomorrow I will prepare these two chapters: elements and biological compounds, and the cell
Yes.
structure and organization.
Exactly. So this is
Okay,
how we'll complete
both.
our component one, and then after we'll go for the component two, right? So because
Okay.
this is last topic here in component one, where just two topics are remaining that we'll cover tomorrow, and then we'll in next week we'll go for the component two as well. But keep in mind somewhere, assessments are quite important. I used to upload some questions on the portal. I don't know you have checked this or not. In last week, I have uploaded a questions for you there. Few of the questions for.
Okay, I will do it.
Yeah, so that's
Okay,
also
I will
fine.
do.
Otherwise, we'll also conduct some assessments from our side as well. Have we prepared all of this or not? So this is how
Oh,
it would help you to, I mean, practice more. So that those questions would be as the same pattern that you have. Suppose we can do one thing like to take any past paper and to solve it, the component one, like I mean the paper one
yeah,
you
yeah.
would say, right?
Entire component one.
Yeah, entire
Okay.
component one. Then go for the component two, and because both have the 50%, weightage, so we can work on that.
Yeah, yeah, yeah.
As far as you should keep in mind, few more things, Abdul. Like I would say, let me tell you one thing more. Um, yes. In case of educators, what I have observed, you should also be able to understand in component one higher. You would see like, uh, you know, uh, how the questions are distributed here. For instance, I would say in case of component one higher, you would have 10% of maths questions. A mathematical skill that will be about 10% of your component one, right? And as per the
Yes.
practical context, that's it's not given in your specifications, but what I have observed in the past papers, what I have analyzed, most of the questions which are um, about 25% to your practical context, like that is given there. Okay, and extended
Oh,
response, as we know, that's about 5% of that. So in component one, which is there, okay. So as far as MCQ's presence, that's
okay.
not fixed. That's not fixed in any of the sections. So this is what we should prepare overall. So component two, we will discuss that as well. That would be, there you would see. in your component to hire, there will be about 10% of your extended response. That would be 5% more as compared to the component one. And in a similar way, the maths portion is about 23%. You just imagine about 25% are missed. It means like in component two, we'll have to work more on the mathematical skill as well. So there, because it's about 20 to
Yeah.
25% there in your component two, right?
Yes.
So in a similar way, practical
Yes.
context, that's about, yeah, that's about 60%. I mean, it is even more than that. So although the practical context is quite less in case of component one, that's only about 25% or 23%, right? So keep in mind while having questions in Educus, we should also understand like either we are covering these different question styles or not. That's very important. Otherwise, you may bring some questions as well as a mathematical skill is concerned, somewhere you were observing. And once we'll prepare or we will just practice on the past papers, so there you will see how these questions are. And as far as here, what I'm practicing right now, I'm taking in my worksheets what I'm using for you. So I haven't included the mathematical skills as well. So right now we have to take some questions from our remaining portion. Hmm, let me check one thing more for you. I can go to do one thing more. Let's just a moment. Um, easy. Hmm. We have covered here. Hmm, yes. Yeah, we're talking about the cell division. And today I'm going to have some questions where we can apply our concepts. Cell divide by mitosis to form new cells. Describe the change that occur within the cell between the beginning of the prophase and okay. So it is a bit question that I'm going to put. Like it's a 5 mark question for you. And what you need to do is, oh, have I shared something? Oh no, I didn't. Just a moment, please. It's a whiteboard. Abdul, look at this. Here's a question. Cell divide by mitosis to form new cells. Describe the changes that occur within the cell between the beginning of the prophase and the end of the metaphase. Um, that's straightforward question, like not much in depth, but yes, we need to work on it. Yeah, what do you think? What type of changes
Yeah.
are there?
First, yeah, the in the prophase, the chromosomes condenses
Mm-hmm.
and they become visible,
Mm-hmm. Okay, good.
and
Mm-hmm.
the spindle fibers forms,
All
and
right.
the chromosomes when they condense, they become two sister chromatids. So when
Mm-hmm.
the spindle fibers form, they form at the centromeres of the chromosome.
Mm-hmm.
So then they and the metaphase, they
Mm-hmm.
line the chromosome at the equator of the cell.
All right. Great. So now you can write as per I will just calibrate this with mark scheme. So we'll check it how to. It's a 5 mark question. Yeah, yeah, definitely. Between prophase and the end of matter phase. So we need to consider
Yeah.
the changes about 5 to 6.
Yeah.
Yeah, I have to add the mark scheme. Yeah, that's fine. It's a full sentence response, okay? Not in points. In prophase, the chromosomes become visible, showing two sisters. Great, good. Yes, good. The spindle fibers from centrioles join at the centromere of the chromosomes. Great. 3 points are here, two more. In anaphase, what are you going to do in anaphase? We are not discussing here anaphase. It's metaphase.
Yeah. Oh, sorry, sorry. Metaphase, metaphase.
Yeah, it's metaphase, not anaphase. Just a minute. All right. Good. Right. The spindle fibers line up the chromosomes, or you would say the chromosomes aligned uh, by the spindle fibers at the equator of the cell. Yeah, that's fine. How many points are here? Three, 4 or 5.
I think these are almost 4 points.
Add envelope breakdown is also there somewhere you didn't mention envelope breakdown. Because that's also the part of change. In case of
Yeah, in
prophase.
prophase.
Yeah,
In prophase, the
nuclear envelopes breakdown, and you talked about the spindle fibers formation and they extend
yeah,
from the centriole.
yeah, yeah, yeah,
Yeah, exactly. The spindle fibers from centrioles join at the centromere of the chromosomes. We should also think like, yeah, that's fine. But one thing more, like, you know, each chromosome consists of two sister chromatids, which are joined at the centromere.
yeah.
That's one more point that you
So,
can add here.
so,
Mhm.
so I can just show right here that the chromosome become visible, showing two sister chromosomes joined at the centromere.
Yes, visible, showing two sister chromatids join at the centromere. Yeah, good. This is how we can correct this. Great. Now it's a five mark question. All right. The diagram below shows a chromosome in a cell undergoing mitosis. Identify P. Describe the role of structure P during mitosis. Okay, it's easy. Although it's I've changed worksheet, but I'm getting back to that as well. Here's a question. The diagram below shows the chromosome in a cell undergoing mitosis. You need to identify P here first. It's where we have just
Centromer.
discussed. Yeah. And describe the role of the structure P during the mitosis. What's its role here? It's actually just
Okay.
relevant to the previous one. Hmm.
I can say holds two sister chromatids together and
And it's any other role.
yeah, with the spindle fiber. I'm writing that as well. Yeah,
Hmm. What's your answer? Hold two sister
done.
chromatids together, joined with
Yeah,
spindle.
done.
Yeah, joined with the spindle fibers. And also, this is a centromere, which actually allows the chromatids to go to the opposite pole. To you would say like um, it actually uh, helpful for the separation of the sister chromatids to the opposite poles as well. Like we know about it. Once they are aligned in the center, so then this is the centromere from where the sister chromatids, they will be separated, they will be detached, and they will go towards the opposite poles. So it means like this is also you would say allowed by centromere. All right, getting Abdul?
Yeah,
Okay, so what we can do here, like suppose I have a graph here. That's I should cover its. Hmm. Okay, just a moment. Question is like I have to choose question from here. That's of about suppose this one. Look at this. Hmm.
so they are saying scientists investigated the effect of meiotic inhibitor drugs which prevents the microtubule formation. One such drug is Vixarin, a chemotherapy drug that binds irreversibly to the microtubules, a spindle protein in S phase, where cell zyga interferes with the formation of meiotic simple. Okay. Explain why preventing the formation of spindle fibers stop the cell cycle. So I can write that
Hmm.
formation.
Preventing spandrel fiber
Oh,
formation means what?
like so the spindle fiber will not join to the centromere, so they will not be pulled at the each end of the equator, and so
In
my
other words,
mitosis will be disrupted.
uh, I mean, we would say like if the spandrel fibers formation is prevented, so we know the consequences. One is like the chromosomes can't attach to spandrel fibers, right? Because um it's prevented, its formation is prevented, right? So they can't be lined up on the equator as we usually see in the normal cells, right? This is also important. And suppose one more thing that I can, what's in your mind for? So first, you should tell me, and then I'll tell you like how we can. Sister chromatids can't be separated, right? And definitely can't be pulled to the
Yeah.
opposite poles, as you said. So in other words, the mitosis can't continue and the cell cycle stops, right? This is for this is actually, I would say, the consequence of the prevention of the spindle fibers formation. right? So 3 or 4 points that we need to add here for 3 marks because it's a 3 mark question. So at least we need to add 3 points. One, we said like chromosomes can't attach to the spinal fibers. They can't line up on the equator. Sister chromatids can't be separated and pull the opposite poles. So mitosis can't continue and the cell cycle stops, right? This is what we should consider. And then after we'll talk about the question B as well. Are you getting Abdul? Any confusion here?
Yeah, no, no.
It's okay. Just write down the answer. Finally, yeah, formation of the, okay, you are right about it. Formation of the spinal fibers occurs at prophase. Inhibiting this process will disrupt mitosis, and these two sister chromatids will not line up
I'm
at the equator, and chromosomes will not be separated and to move to the opposite
yeah,
pole.
yeah. I'm saying two sister chromatids will not be separated
Yes,
at the during anaphase.
yes, during anaphase.
Yes. So now question number two, explain why the medicine can be used as treatment for cancer even though it affects some healthy cells.
Hmm. Yeah, this
Because
is Winston.
it, uh, it will
Hmm.
control the, I can say, the uncontrollable cell division. No, no. It will control, yeah, uncontrollable cell division. This which happens in cancer.
In what sense? Like,
Or rapid cell.
hmm.
It will control the uh, division of cancer cells.
Hmm. Why Vincertine can be used as a treatment for cancer even though it affects some healthy cells. You know what? We need to compare cancerous cells and the normal cells. As we know, in case of cancer cells, their division is uncontrollable and very rapid as compared to the healthy
Yeah,
cells, right? So,
yeah.
we'll talk about it like it is always given in the form of some doses, once every 3 weeks. We'll talk about this is another question that is there in option in C. When Christine was given to a cancer patient, it was given once every 3 weeks, starting at the time, oh, like zero. So we'll see like why we're going to do this, right? So again, you would say, the cancer cells have the very rapid cell division as compared to the healthy cells. So that's why I would say like this drug is used in order to um, somehow to control the cell division in cancer, right?
Yeah, yeah.
So all we'll see like these healthy cells are also affected by it, but cancer cells are more affected because their division is more rapid as compared to healthy cells, right? And
So I wrote like it will slow down the uncontrollable cell division of cancer cell as the cancer cell divide more rapidly than healthy cells.
yes, although the healthy cells are also affected because of this, but because we know the cancer cells, they are even more rapidly dividing, and definitely this drug is playing a critical role to control
But
those cells
like
division.
if we imagine like this, so like two type of cells are dividing. One is dividing more rapidly, and another is little bit slow. But
Hmm.
when we put this drug, the cell that is dividing even more slowly, like a normal body cell, will be more effective because it will be about to it will no longer divide because it will be compared to the cancer cell. It will still be dividing. You know what I mean?
Are you saying like this drug will affect the normal cell even more than the cancer cells or prevent its division?
Yeah, the the division of the muscle, for example, muscle cell
Hmm.
is already much slower than the cancer cell, so it will be even more affected.
Yes, affected, but our problem is right now because the cancer cells are more there, and they are our main target is to control them to prevent their division further, right? And in the next question you will see like the effect in normal cells is repairable. That can be repaired. How? That's why we are not giving this
Yeah,
this drug
through stem cells.
frequently. In next question you will
Hmm.
see your question or your confusion will be cleared in this next question. What is this? What's the next question? When Christine was given to a cancer patient, it was given
Which
once
Christine
every
was
3 weeks,
giving.
starting at the time
Hmm.
O. This is what we need to answer this question. Let me put the further like its remaining part from the worksheet. Suppose we have this one like this. So again, definitely we need to
Hmm.
answer this question. The previous one, C. Um, suppose using this graph, calculate the number of cancer cells present at week 12 as a percentage of the original number of cancer cells. This is a 3-mark question, but again, in previous one, we can see like we always give this drug once in a 3 week. It means like it allows time for healthy cells to recover in between the doses, and while still
Whereas
preventing the cancer
I think
cells
the
from
cancer cell
dividing.
will take more time to recover.
Yes. However,
And-
the healthy
Oh,
cells will
yeah.
be
But the
recovered.
cancer cell will take time to recover. Yeah.
Exactly. So healthy cells will be
Yes.
recover between the doses that we are providing, and they are even definitely they are rapidly recovering as compared to the cancer cells. But cancer cells, they're still preventing cancer cells from dividing. The drug is more effective there on the cancer cells, right? So this duration between the dose injection or the dose delivery is like it provides some time to recover by the healthy cells, right? Getting?
Yeah, now they are saying the calculate the percentage of the cancer cell present at week 12 compared to time 0, week 0.
Hmm. Yes.
So,
Hmm.
so I can 0.4, 0.4 times by 100,000 are present at the end of week 12
Hmm.
compared to there are 500,000 present,
At week 0, the number of cancer cells was about, you can see here, 2
500,000
multiply 100,000. At week 12, the number of cancer cells was about zero point 0.1 into 100,000. That's the difference you can see here. And if you want to go to have a percentage,
Yes.
definitely obtained over the total that you have to multiply with 100. So 2.0,
It is 0.08%.
how? How 0.08%? Hmm. How you can see?
I'm I'm dividing the number of cells present at week 12,
And
which is
what's the num
zero
what's
point
the
0.4
0.4?
hundred times by, and the
Okay.
original is 500,000. 500,000.
At week 0, the number of cancer cells was about 2.0. Here it is. Of into definitely we'll multiply it with the 100,000, right?
No, they're saying at the time of 12 weeks.
But you have no. I'm talking about the
No, they're
first one.
saying at the time of 12
I,
weeks. So we
I'm
will compare from the
I'm
Z.
talking about the at week 0. At week
Yeah.
0, the number of cancer cells was about 2 into 100,000. 2.0 into
I think
100,000.
no, it was one. If you see zero, it is
Hmm.
there are 500,000. The line is at five.
At week 0.
They are, yeah, if you're
Mm-hmm.
saying first dose, look, the line is at five. So the graph is starting from five.
Hmm. The first dose. Okay. And the graph shows the changes in the number of healthy cells and cancer cells during the 12 week of treatment. First dose that you have provided, using the graph, calculate the number of cancer cells present at 12 week 12 as a percentage of um you're not you need
It's
to
8%. It's
yes
8%. It's 8%, I think.
Abdul,
No.
listen. Once you have provided the dose, that's the start, right? And you can see this graph is going down. This line is coming down. The reason is why, because it is actually preventing the healthy cells as well. Like at the first dose, you can see in one case, uh, like I can show you here in this case if you understand it. Let me show. Oh, just a moment, please. Where is this?
Is it not the answer 8%?
Uh, no,
The answer is not 8%.
no, not 80%. Not 80%, but before that, we need to dissect it. What does it mean? You're starting it from 5, right? This one. But I'm saying if you have provided this dose
Yeah.
1, it is going up to
Yes.
this point. This is the decline
Yes.
that we have to see, right? Here you have provided this dose. This dose is effective in the healthy cells up to this point. Getting or not
Yeah.
in one case, right?
Yeah.
And that's why I'm saying like the number
No,
of
they are
cells.
only saying asking about the cancer cells.
But you have to take like okay, the cancer cells. Using the graph, calculate the number of cancer
They
cells
only
present at week 12. But we have to take, the total number is again 1,000. What you need to do is we should consider at week 0, okay, the number of cancer cells was about hours. Okay, not healthy. If this is for cancer cells only,
five
so at week 1, the cancer cells was about
500,000. 500 500,000. At week zero. Original number at the
just explain it how. Just explain it for me.
no, they
Uh,
are saying calculate the number of present cell
yes, cancer cells
at week 12 as a percentage of the original number of cancer cells. So when you go original when we go at the time week zero and we just follow the graph on the y-axis, the line will be at five, not
yes,
one. One is
yes,
at
you have to get back to the no these week 0. Hmm? So there, if you are going to find the
so
percentage, yeah, it's fine. Yes, I was thinking like so asking about the healthy cells as well. Like you have to compare the. So it's only for using the graph, calculate the number of cancer cells present at week 12 as a percentage of the original number of cancer cells. That's fine. So this is actually clearing this point. Yeah, this one.
Yeah, so I already solved it. It's 8%.
Uh, yes. But suppose somewhere the question is for healthy as well. So what you will do in that case?
Like what if they ask like what like if they ask like the what they ask, what they will ask, it will be according to the question.
You know, the one, the type one is, I mean, this line is showing you healthy cells. And can we see any
Yeah,
change in second dose, third dose, and fourth dose in healthy cells? Any of the change there?
yeah, they are decreasing. Yeah, they are decreasing as well. So we can find the percentage of decreasing cell as well.
We can
It's
find out.
2.9.
Hmm.
2.9
Hmm.
times by 100,000 divided
Hmm.
by 500,000
Hmm.
multiplied by 100. So that we
100
will get the
multiplied by 100.
percentage.
As we know, like
Yeah.
the formula is simply we know that we always use the for the percentage that's obtained over total that you have, and you have to multiply it with 100. That's fine. Nothing is much difficult in this case, right? So
Yeah.
again, the graph is important. Like where we can, if we can extract data from here, in any of the case, like whether that is for healthy or for the cancer cells. So somewhere they may ask you for the healthy cells as well, but definitely it's for cancer cells. This question is specific to,
Hmm.
right?
Hmm.
So then after we
Yeah, नहीं
have
that's
the
नहीं
what's the next question?
निकलेंगे.
Describe to differences in the effect of Wins uh, Christine on cancer cells compared with the healthy cells. Yeah, this is the second question which is there. Is it pasted here or not? No? Okay,
No.
let me take this.
कौन सा
It's for 2 mark question, right? Abdul,
गिरा हुआ था मुझे.
like you have a 2 mark question here, and it is now you need to compare it with the cancer cells. Somewhere I've seen this any of the past papers where they asked about the healthy cells as well. So that's why I was thinking like this is that specific question. You can see here, describe two differences in the effect of vincristine on cancer cells compared with the healthy cells. Now we need to compare it, right?
Yeah. The
Hmm.
the the number of cells, the number of
Hmm.
cancer, yeah,
Hmm.
cancer. One second.
Good. Hmm. Just compare it by looking at the graph.
Cancer cell will will
Cancer cells.
decrease at the higher
No, no.
rate.
First need to look at the graph. This is showing you what? The first important thing that it's showing you like the cancer cells are affected more strongly, as we have already discussed in a previous case, like because they divide more
Yeah, that's
rapidly
what
and
I'm. Yeah,
continuously, right? So,
this is what I'm writing that the yeah.
okay, okay, right down. It's for 2 mark, okay? As per mark scheme, you need to add 2 points.
One point is, and second point is that the
You need
healthy.
again, you need to contrast with the healthy cells. And healthy
Yeah,
cells are
I have
less affected
no
and
that I
can
have
recover
the
between
first
the doses of
I
the
have
drug.
contrasted.
Okay,
Yeah, my second point is going to be this recovery one, and the first point I have already done with contrast to healthy cells.
okay, all right. That's fine. Okay, good. Hmm. Yeah, rapid recover. Less affected and can recover between doses of the drug because we are giving time once in 3 weeks. Good. Another graph is there where we need to use our concept.
Done.
Done. Let me check it. The number of cancer cells will decrease at the higher rate compared to the healthy cells. The healthy cells are less affected and more rapidly recovered after each dose compared to the cancer cells. Good, that's one. Perfect. That's a good answer. All right. So then after I'm going to have one more graph for you to understand how we can deal with this. Suppose it's a one mark question here, 1 mark for first and 1 mark for the second. Abdul, this one. You have rolled 2 marks.
Okay. Let's see. The graph below shows the mass of DNA during the cell cycle. Using your knowledge of the cell, explain the shape of the graph between 5 and 8 hours. I will say that this process is interface where the mass of the cell, where the organelles replicate.
Hmm. Okay. Right. So how we can answer in 5 and 8 hours. Let me check it for you. Hmm. According to mark scheme.
Done.
5 and 8 hours. This is interface where the organism circulating chromosomes replicate, so mass of DNA doubles. Explain the shape of the graph between.
Now they
Hmm.
now they're saying 13 to 16. This
Hmm.
is where I think
You
this is
even
going to be
interface is
telophase.
uh Abdul. We can more specify this here, like
Hmm?
synthesis phase. In interface, like we have other components as well. So
Yeah,
five
yeah.
to 8 hours,
This
DNA
is the
replicates
S1.
this S phase, right? S phase. So DNA mass increases there, right? That's what we can add. And 13 to 16 hours, as we said, like the mitosis occur, right?
It's
And
telophase.
sister chromatids separate and DNA is shared into two nuclei. Yeah. And so I would say the DNA mass per cell halves, right? Good. What's the next one? Good.
Actually, where each sister chromatid
Hmm.
uh,
East sister
is
chromatid.
located in two identical cells.
I mean, I would say like we should consider the DNA mass per
Two
cell
sister
halves.
chromatids.
Hmm.
So the two sister chromatids are shared in two identical cells,
Yes,
or
two cells. And not we would say identical, but two cells.
daughter cells.
Because we are focused on the mass of DNA. That's our main target. So the graph below the mass of DNA during the cell cycle. So that's why we said in case of S phase it becomes double, and in the telophase, and just after telophase, cytokinesis. So there two nuclei forms and the mass becomes uh, halves, like DNA become halves, right? Yeah, good. Done, Abdul?
What? Yeah, I have already. Oh, next question. No,
Yeah,
actually, I'm pasting the working, which the percentage working. I just one sec.
okay. Apart from an increase in the mass of DNA, give one process which occurs during hours 0 to 14, which enables nuclear division to occur. What's that one process?
0 to 14, which enable interface.
No, no, question is just look at the question. Apart from an increase in the mass of DNA that's going on, give one process which occurs during hours 0 to 14, which enables nuclear division to occur. On the one hand, you can see like the mass of the DNA is increased in synthesis phase, right? But what's the other process which is going on there, and that is actually helpful for the nuclear division?
Cells. The cell organelles replicate as well. The replication of cell organelles,
We need to specify it more. Like spindle operators, that's
like
simple.
chromosomes.
Because spindle fibers and the spindle operators are the microtubules synthesized, right? Centrioles
Yeah.
duplicate. This actually enable the chromosome separation in the mitosis. This is what we should consider, right? So yes, these are these structure, the subcellular structures which are present in the cell, but we need to specify it because spindle fibers and the spindle operators is quite important in this case, right? Getting centrioles
Yeah,
duplicates, it
I'm saying so
contributes.
it contribute in the
You
separation of two sister
microtubules,
chromatids.
microtubules. Yes, you can also talk about the microtubules as well, or the spindle
I'm
operators.
saying,
Because two main important processes which are here, like not two, it's one, but we consider like the two components which are synthesized here. Centriole duplicates. Yeah, great.
done.
It contributes to separation of two sister chromatids during mitosis. Can we add somewhere like to about the microtubules? Can we talk about it? Centrioles
Through
duplicates.
I can say
How we can add
through
this term, this word specific spindle operators? Yeah, it's fine. Centrioles duplication is also like somewhere. It's fine. Answer is fine because it's for only 1 mark, so we don't need to go for spindle fiber formation or the spindle operators formation. Whether you can add that one or this one, it's fine. Any of the two, at least you should know how to use this, right? So this is for the question that we have done with this. Okay. Then after we can go for a few more questions which are related to this. Like we have 4 mark question, and let's talk about this. Here is Abdul. Look at this. A sample of tissues taken from a liver biopsy was analyzed. Some of the cells had 6.4 units of DNA, others had only 3.2 units. Explain the difference in DNA units of these cells. A sample of tissue
so I
taken
can say
from the
that
liver biopsy was analyzed. Some of the cells had 6.4 units of DNA, others had only 3.2 units. Explain the difference in
so
DNA
I can say
units of these cells. Yeah.
cells
Yeah.
with
Yes, Abdul.
6.4 units.
Hmm.
1 second. 6.4 units will be under the process of cell division,
Hmm.
where the DNA replication happens before the mitosis or meiosis.
Hmm. Yeah.
Done. I can also during interface
No.
at inter.
Okay. Yeah. So now B.
One of the cells from scroll down. One of the cells from the samples was analyzed. The
Hmm.
time required for the cell to complete one full cell cycle was
23
23
hours.
hours and 40 minutes. Yeah.
Okay.
Calculate the time required for the cells to produce 8 cells.
Hmm. Means like you have to 1 to 2, 4, and 8. Like this is what's going on.
So two cycles. Basically, we need two cycles.
2 or 3. 1 to
Yeah, and
2,
three.
2 to 3, or 2 to 4, and then 8. So 3.
Yeah,
3
three.
cell cycles are there.
So we need
Getting?
to multiply.
Yeah,
So
exactly.
basically, we need. So I'm the calculation type person. I'm doing on the paper. Then I
Okay.
will
Okay.
upload the image.
All right. All right. That's fine. You can also convert this into hour, like 23 hour 40 minutes. So if we convert that into the hour, so that would be 23.67 hours. Like this is how it's going. Getting? Okay.
Yes.
Okay, all right.
Just 1 second. Just let me.
Okay, okay, all right. All right. Hmm.
Uh, okay, so we need like 3 cycles. So we will multiply 23 hours by 3, then minutes by 3.
Hmm.
So is it 71 hours total?
Yes, it should be. Hmm. About in my the mark scheme that let me check the mark scheme. 71 hours, yeah, in 0 minutes. It's about 2 mark questions, so yeah, that's fine. But one trick is like you need to convert that into hour and then multiply with it. If you know the total cell cycles are there, so it's easy to convert. All right, Abdul, I think let's conclude this for today. And we have 3 4 minutes, but um, then after Abdullah will join the class. And suppose how many questions that we have here. Mm,
So tomorrow I will prepare the biological compounds as well as a cell structure topic as well.
cell? Yeah, the first two
Structure and
yes,
organization,
first two topics
both.
that you need to work on. This is what
Yeah,
I'm
yeah.
giving you for an assignment. Just a moment, please. Meanwhile, you can go for just to say just 1 minute. I just want to add something for you. You can work by yourself on this. Suppose, oh, sorry, what I'm doing. Okay, this one for you. You will work on this by yourself. Have you added here? Yeah, 71. Okay, fine. Great. So this is a question as an assignment for you, and
Are you pasting the questions on the whiteboard or on the
on
portal?
on the board right now on the board,
On
but
the portal or the
because on the
whiteboard?
portal I will add tomorrow because tomorrow you will your last class with me, so I'll add
Yeah.
there tomorrow. Right now I'm just adding here on the board so you can work on this just after
Yeah.
class, right? By yourself. So just one more thing. The sample taken biopsy. Yeah, this is what we have already done. And mitosis, process of living organism, gifted reasons why mitosis is important. Easiest, I think. Diagram is already given there, so look at this. Okay, I've pasted here. All right, that's it for that's all for today, and we'll continue this tomorrow. So meanwhile, you can do these two questions, attempt two questions by yourself. And tomorrow we'll talk about two topics from the component one, and then after we'll complete our component one, right? We can also have overall assessment on that just by attempting to. But I would suggest you before going to the component two, what we should do like to attempt few of the past papers um as a whole in all of the I mean specific to the
We
component
can do a
one.
couple of past papers.
Yes, couple of past papers for the
Yeah.
component one, and then after we'll go for the component two,
Okay.
right? Okay, all right. So that's all for today, Abdul.
Okay.
See you tomorrow. Take care of yourself. Bye bye. And ask
Uh,
Abdullah to join. Abdullah is already in the class.
uh,
Bye bye.
Okay,
Bye bye. Bye bye.
bye bye. Hello, Abdullah. How are you?
I'm good. How are you?
I'm fine. What's
I'm good.
about
How are
you?
you?
Yeah, I'm fine, Abdullah. What's up? How was your day? What's going on? Tell me about something that's special
I'm
for today from your
that's
side.
good.
What you have done that was quite special for you. Nothing?